Welcome to the first installment of Episode Genetics. My name is Daniel Crossman, and I will be your host today. So, the first thing that we are going to do is we are going to jump back in time, specifically to 1847 to meet this fellow named Rudolph. And Rudolph Virchow, um, is a very interesting guy. Did a lot of stuff with pathology. And Rudolph in 1847 is living in Berlin, Germany. And he had just coined a new term, leukemia, which, if you can do a little bit of linguistic investigation there, is going to translate to leukemia. So we're going to jump in our time machine, and we are going to go all the way 163 years into the future to 2010. At that point, I am sitting alone in a hospital bed in Virginia here in the States. And I had not been feeling well for the entire summer, out of breath, fingernails turning purple, wanted to sleep all the time, and doctors just did not know what was going on with me until a hematologist got a hold of my records. I just finished getting an enormous amount of blood in the form of a transfusion and came in to tell me that I had acute lymphoblastic leukemia, or ALL. Now, what is ALL? Acute just means that it can progress very quickly, and if not treated, it'd probably be fatal. Lymphoblastic, another word here, another way to say this is acute lymphocytic leukemia. And all that means is that it develops from lymphocytes, which is a type of white blood cell. So the difference between leukemias and other types of cancers is leukemias tend to form, or they do form, in the blood. They tend to form along the white blood cell path here, although they can form along other pathways. But for me, the lymphocyte is our culprit here. Very, very important in our immune system, obviously, and you can see that it goes on to create some really vital components to our body. So the difference between ALL and something like esophageal cancer is this cancer is starting in the bone marrow, where esophageal cancer could start, obviously, in the esophagus from like an environmental factor, perhaps. Obviously, in the bone marrow, there's already rapidly dividing cells in a normal functioning human. So when cancer is introduced, then it just gets out of control and overcrowds the normal healthy cells, which is why I needed blood. I just simply wasn't creating healthy, normal blood. So why is ALL important? To me, I think looking at the statistics makes the most sense here. It is in comparison to other cancers here in the state, it's actually quite rare. Of all the cancers diagnosed in the states each year, it accounts for less than 1% of all those diagnoses. However, of all leukemia diagnoses for those under 20 years old, it accounts for 74%. Now, that under 20 years old is kind of important because this tends to be a childhood disease. Although, I mean, I was 20 when I got it. There are people definitely older than 20 who developed this as well. The 5-year survival rate, which just means after your diagnosis, how likely are you to survive past 5 years, is shown below there. If you're under 20 years old, you have an 89% chance. If you're over 20 years old, it plummets down to 38%. Interestingly, you are much more likely to develop ALL if you're younger than 5 years old. Also, if you're over 50 years old. Kind of interesting. Males are affected slightly more than females, and they aren't sure why. Males also require longer course of treatment. So I had 3.5 years of chemotherapy, where females would be closer to 2 or 2.5 years. Okay, so this is a genetics class. Let's talk a little bit about genetics, inheritance, and some risk factors or causal factors. Unlike some other cancers, maybe something like breast cancer, inheritance really doesn't come into that big of a role with ALL. It's not unheard of to inherit acute lymphoblastic leukemia, but it's just not very common. Um, my parents didn't have ALL. My grandparents didn't. I was the first one to have this, and so that means that mutations are developed during the patient's lifetime typically. And scientists just aren't completely sure what exactly causes leukemia in general. For sure, there's no big smoking gun, but there's a lot of like risk factors and causal factors that can go into it. Some are shown here. So, like, one idea is low level radiation as a young child. So, like X rays and CT scans, although this isn't totally well understood yet. Chemical factors, specifically benzene, which is widely found. It's naturally occurring in stuff like forest fires and volcanoes. Can't say I was around a whole lot of volcanoes as a kid. But it's also man-made. It's in, I mean, plastic, gasoline, hair dyes, you name it. One of the biggest culprits is cigarettes, cigarette smoke. Again, wasn't around that as a kid really at all. There are also viral infections as a risk factor. So, human T cell lymphoma, leukemia virus 1, HTLV1, and the Epstein Barr virus, which here in the States most often causes mono, which would help to explain that why they thought I had mono before I received this diagnosis. You also have genetic risk factors. So, if a person has Down syndrome, Klinefelter syndrome, Bloom syndrome, just to name a few, that they're at a higher risk for ALL. Okay, so mutations. So you can kind of see the outline here. Um, Here it's talking about oncogenes, which are really important. They help the cells divide, among many other things. And if those get mutated, then those can lead towards cancer cells. Another cell that's commonly found here is tumor suppressor genes. And those do exactly what they sound like. They keep the cell growth and division under control. So if those get mutated, then it can lead towards cancer. So in ALL, the biggest culprit is translocation for a type of mutation, and specifically something called the Philadelphia chromosome, which is a swap of DNA between chromosome 9 and 22. Of course, there are other translocations that have been found, like T411 or T814, but this Philadelphia chromosome is the one that's most well known. There are other things that go into play, like um, deletions, just a loss of genetic code, or inversions. Those are important too, rearrangement of DNA in a section of chromosome. But mostly it's that translocation. And in leukemia, a lot of times scientists just simply do not know what genetic malfunction happened. Still a lot to learn. In fact, I was quite surprised at how little we know about the risk and causal factors involved with this. In future episodes, we'll dive deeper into genetics. Probably going to go more into like the Hox regulatory pathway, translocation involving the MLL gene. Might do some stuff on like the chimeric transcription factors, all play a role. So I hope you enjoyed the episode. And from me and Rudolph, we'll see you next time.